Before scientists can grow malaria parasites in the lab, they first need something very specific: healthy red blood cells. In this video, I'll show you how we isolate and wash red blood cells from whole blood so they're ready for culturing the malaria parasite, Plasmodium falciparum. But this technique isn't just for malaria research. Purified red blood cells are used in many areas of biology and medicine. Let's start with the basics. Why are red blood cells needed for malaria research? The malaria parasite, if you remember, and I've got a video on here on the life cycle of the parasite, the malaria parasite spends most of its life cycle inside red blood cells. That means if researchers want to study parasite growth, drug resistance, or invasion biology, they need a reliable supply of healthy red blood cells. But whole blood contains much more than red blood cells. I have a video on here about what the components of blood are. If you remember, there's plasma, white blood cells, platelets, and other components. So you can have a look at that video to go deeper. So before we can culture parasites, we first need to purify the red blood cells. We start with whole blood collected in an anticoagulant tube. Two common anticoagulants used in blood collection are EDTA and heparin. In our case, the blood was collected in ACD tubes, which stands for acid citrate dextrose. Anticoagulants prevent clotting and keep the blood cells separated and usable for laboratory work. The first step in separating the blood components is centrifugation. This is where you spin the blood and it separates into components based on density. You typically see three layers. The top is plasma. Because we used an anticoagulant, we call it plasma. If we hadn't used an anticoagulant, it would have been serum. The middle is the buffy coat. I also have a video on here where other researchers may be interested in studying immune cells. So they're interested in peripheral blood mononuclear cells. Then finally, the red blood cells are at the bottom. The first thing we do is remove the plasma, which is the yellow liquid portion of blood. Plasma contains proteins, antibodies, and other molecules that we don't want in our malaria cultures. Next, we carefully remove the buffy coat. See, this is the white blood cell layer. Then this layer contains the red blood cells as well as platelets. Removing it is important because white blood cells can interfere with parasite culture and experimental results. At this stage, we're left with mostly red blood cells, but they still need to be washed. So we add incomplete RPMI 1640 medium to the cells. The cells are mixed and centrifuged again. This washing process removes remaining plasma proteins and contaminants. In our protocol, we repeat this washing step 3 times. Each wash improves the purity of the red blood cells and ensures they're suitable for malaria culture, for parasite culture. After the final wash, the red blood cells form a clean pellet at the bottom of the tube, and we use this with complete RPMI. So we add some lipids and fats in the form of Albimax or human serum. And you actually want to resuspend the red blood cell pellet with the complete RPMI that has the lipids and fats added so that it's at 50%. So you use equal volume of your pellet to equal volume of your complete media. To do the culture, the final culture, we want the red blood cell component to be at 5%. So what we do is actually just make a 5% solution. And then when we come time to culture it, because the cells are typically cultured at a 5%, what is referred to as hematocrit. There's also another video on here on how to calculate your hematocrit. You basically say you want it to do a 1 in 5, then you just take one part of your parasite culture, the previous culture or ongoing culture, into a new flask that has 4 milliliters of the 5% hematocrit blood, and that way you maintain that 5% hematocrit. When the parasite infects these red blood cells, it can grow and reproduce inside them, allowing researchers to study its biology in controlled laboratory conditions. So although we used this method for malaria research, purified red blood cells are used in many other areas of science. For example, in transfusion medicine, scientists isolate red blood cells to study blood group antigens, like those in the ABO blood group system. I also have a video on here on the ABO blood group system. This helps determine whether blood from a donor is compatible with a patient. Researchers also isolate red blood cells to study diseases that affect them, such as sickle cell disease. Purified cells allow scientists to examine cell shape, membrane structure, and oxygen transport. And malaria isn't the only parasite that infects red blood cells. Organisms like Babesia microti also invade red blood cells, so similar purification techniques are used in those studies as well. So in this video, we walked through how to prepare red blood cells from whole blood for malaria culture. We started with anticoagulated whole blood. We centrifuged it to separate the layers. We removed plasma, we removed buffy coat, and then we washed the remaining red blood cells 3 times in incomplete RPMI. Then these purified cells were resuspended in complete RPMI, which can then be used to culture malaria parasites and study their biology. If you're interested in more laboratory techniques used in malaria research and biotechnology, you can consider subscribing to Ajua Biotech, where we can break down complex lab methods using simple visuals. I have linked all the videos mentioned in the description box. Thanks for watching, and I'll see you in the next video.